that happen? Well, let's take a look at two mothers, okay? One of them had 10 children in 17 years. Peter's seen this slide before. I got it from David Haig. 7 of them survived. These were the interbirth intervals. Look at how short some of them were. She was having babies 1 year apart for part of this. The last one took a bit longer. She started having babies when she was 31, and she stopped when she was 48. Here's another mother. Oh, she died when she was 88. This other mother had 9 children in her. She started when she was 13, and she finished when she was 44, and she died when she was 46. Any ideas on who these two ladies are? Yeah? This or that? Yeah, okay. You got it. This is Emma. Okay, that's Charles Darwin's wife. And this is Fifi. Okay? She's on the right. This is what Emma's reproductive schedule looks like, and this is what Fifi's reproductive schedule looks like. And look at how tightly packed those guys are. And look at the fact that they end just about on the same date. What's happened is that Fifi has gotten more in by reproducing earlier, and she reproduces until she dies, whereas Emma has menopause and a long post-reproductive interval. So she started late, produced babies rapidly, and Fifi actually is an outlier. She is actually much more human-like than most chimps. Most chimpanzees do not have this many babies. So if you look across humans, bonobos, chimpanzees, and gorillas, and you put down the major life history traits, weight, adult weight for males and females, the ratio of adult and female weight, gestation length, birth weight, age at weaning, and so forth. The things that really stand out are our babies are heavy, they are weaned young, we start reproducing at later ages than our closest relatives, we live longer, and we have a shorter interbirth interval, although our interbirth interval, in fact, on average, is not really that much different than bonobos. I've put up here in red the Pearson correlation coefficients for all of the traits except the ones where there's data missing. And you can see that actually we're 99% similar to bonobos and chimpanzees overall, and we're about maybe 10 or 12% different from gorillas, if you take that as an overall measure. So you can see the evolutionary thinking that's going on here. We all shared ancestors. We shared ancestors with bonobos and chimps probably 5, 6 million years ago. We have changed since then. Why did we change? We'll get into some of the why, but in fact we don't know all of the why. And what I'm now going to do, and try to do this fairly quickly, is just march through the human life cycle, starting with the gametes, and show you some of the evolutionary issues that come up at each stage of our life cycle. So when you ladies in the audience were 3-month-old embryos or fetuses, you developed your ovaries and you stocked them up with about 7 million oocytes. And between that point, they were progressively eliminated. By birth, you had about 1 million. Chris told me that by menarche, it's not really 1,500. It's more than that. It's like 400,000 or something like that. Yeah, 400,000. So I need to correct that figure on the slide. But the point is it's going down, and it's going down strikingly. And so the question is, why make a lot and then kill a lot of them? And there is some evidence suggesting that the genetic quality of the oocytes increases during the process. So if you just manage to recover fetal material and do DNA sequences on, for example, mitochondrial DNA, the later that material is collected, the fewer mutations you find. In other words, the ones that had the more mutations are being eliminated. On top of that, this is actually difficult data to get. You have to have very close monitoring of conception times and things like that to get it. But 60 to 70% of concepti are aborted in the first menses. That seems to be the best estimate of that. Yeah. that's part of the aging of oocytes that goes on. So if we think about age and size at maturity, so the difference in oocyte quality that you get, say, between 8 years of age and 16 years of age, you're probably getting better and better oocytes. But what happens then is that there's aging of the screening mechanisms. So the screening mechanisms don't get as they start to fall apart, and then as you get older, you're more likely to have a conception with a damaged oocyte than you are when you're younger. Okay, that's another process. Carol Ober's done a lot of work on recurrent spontaneous abortions in Hutterite women, showing that the human female reproductive tract discards concepti that are immunologically deficient because the father's MHC alleles, the alleles that are, well, the MHC and some other immune genes as well, are too similar to the maternal ones for the immune response to function. In other words, those women are throwing away babies that would probably have died of an infectious disease in infancy. We also now know from ultrasound that about 70% of conceptions that start as twins end as singletons. Okay? So that means, by the way, that there are a fair number of singletons who are mosaic in the sense that they're genetically mosaic. They have some tissue or some blood type from the twin early in gestation. Their brother or their sister died and was reabsorbed, and they carry the signature of that with them for the rest of their life. So this is all evidence that shows that the human female reproductive tract has been designed by evolution to function as a quality control mechanism. And by the way, that puts a big filter on mutations. And if you look at where do the mutations come from in humans, they come mostly from men. About 10 times as many of the mutations that humans accumulate originated in the father as in the mother, and that's because this whole process has been screening them out. They don't get into the next generation. Okay, a little bit about size at birth. We have this range, about 1 to 4.5 kilos. We've got larger brains and are fatter than babies that are born to chimps or bonobos. Interestingly, relative to our adult brain size, if you look at how big the brain size is of a newborn chimp versus an adult, our baby should have even larger brains. But development's constrained by the diameter of the birth canal, as Chris pointed out. This is one of the big trade-offs in human reproduction. And that is why babies continue growth, brain growth and differentiation for a long time after birth. And our average birth weight is about half a kilo less than the optimal one for infant survival. And this could be either because of the trade-off with the diameter of the birth canal, or it could be due to parent-offspring conflict. And we won't go into conflict theory very much here, but that's another possibility. Now, ideas on why we have such fat babies. Anybody got an idea? Yeah. Yeah, we need to myelinate our axons, right. And we have a period of really rapid growth, and our ability to rapidly develop an organ that will be essential in our social life and in our learning ability evidently has become a top priority in humans. And that's certainly one of the good reasons that our babies are so fat at birth. And we know that low birth weight adversely affects survival, health, growth, and mental development. Peter Gluckman is going to talk a lot about that. So right at the beginning of life, both just with gametes and with how big the babies are and in what shape they are when they're being born, we can see that evolution is operating and is setting up the phenotype for the rest of the life. We have interesting growth patterns. Okay? Human growth is target-seeking and self-stabilizing, so that if you grow slowly at one point, you'll compensate and grow rapidly later to try to hit the same target. The target is not fixed. It's a reaction norm, but that happens anyway. Unlike other organisms, we actually have kind of a complicated period before adulthood. We have infancy, childhood, a juvenile period, adolescence, and then adulthood. And the thing that's really different about humans is the prolonged length of childhood. chimpanzees tend to go from infancy fairly rapidly into a juvenile state. We have prolonged that childhood state, and this is where that additional brain growth occurs and that fat is used up. It is thought that childhood may be a feeding adaptation that allows shorter interbirth intervals. After all, childhood is when the baby, the infant, can now toddle about, and perhaps it can be cared for by other individuals in the group, and mom doesn't have to breastfeed it anymore. And that may be one of the co-adaptations that led to shorter interbirth intervals. We have a uniquely large adolescent growth spurt. I'll show you the growth spurts in a sec. Interestingly, girls look like adults and can have menses, signaling that they are reproductively capable before they're fertile. It's usually about a 1 or 2 year period. And they can engage in adult roles without becoming pregnant during that 1 or 2 year period. This is actually the basis of, the observation that, say, at a liberal arts college, the females are women and the men are boys. Boys can produce functional sperm before they complete growth, so they are the opposite of girls that way, and they develop before they develop adult appearance. Okay? So they are actually reproductively capable before they're sexually differentiated. It's, again, a 1 or 2 year effect. And I just wanted to throw this in. This is from Chris Kuzawa. This is apparently true for boys in the Philippines that he's studying. Baby boys who grow faster in their first 6 months of life mature earlier, larger, and have more sexual partners early in life. So there can be direct early life effects on reproductive success in some human populations. Here are the growth spurts. Okay, so babies start off growing really rapidly, and then in childhood, the growth rates tend to stabilize. Then as you get into adolescence, the female adolescent growth spurt occurs earlier than the male. It's offset by 3 or 4 years. This leads to things like the girls being the tallest kids in the class in 7th grade, and the boys then catching up and passing them by the time they're seniors in high school. I've gone over the advantages of early maturity, that's short generations, and the security of reproducing before mortality strikes. In humans, the advantages are not only increased size, but knowledge, experience, how to get a hold of resources, how to navigate the social system, all of that kind of thing. They all help babies to survive. Then, of course, the proximate thing that's going on is the hormonal control of puberty from the brain, and that is also influenced by environmental cues. Peter Ellison knows an awful lot about this. So nutrition and actually the presence or absence of a father in the family group can influence that. And as you've seen, this is actually the proximate mechanism driving the production of the evolved reaction norm. Puberty is completed earlier under better nutritional conditions and with increasing obesity. And importantly, age at puberty correlates with age at first birth. So you can see how the hormonal control of this differentiation of the secondary sexual characteristics correlates with the production of the reaction norm. Now, what's going on currently? Well, we are maladapted. We are so well nourished that biological puberty is occurring earlier and earlier. Psychosocial maturation and socially accepted age at first birth are increasingly delayed. This increases the discrepancy between the bodies and the cultures of adolescents. So they're getting conflicting messages from their bodies and from their cultures about appropriate behavior. And that leads to all sorts of issues. But I think that you've seen by now, going through the arguments on the evolution of age and size at maturity and some of our differences from chimpanzees, that this sort of social problem, with all of its attendant juvenile delinquency and potential for crime and teenage pregnancy and all that kind of stuff, is actually set up in an evolutionary framework and with a mismatch to modernity. Peter Gluckman, the other Peter in the faculty, has actually been advising the government of New Zealand on life history theory in terms of how you deal with some of these social problems. And he tells me they've been listening. This is just to give you that graph that I showed you over there. These are the actual data on infant mortality rates in the United States based on over 100,000 births back 1960-61, showing you that, in fact, if you do mature earlier, you are paying a price in terms of the probability that your child will survive. And it gets pretty bad pretty quickly. Okay, now, earlier I showed you a reaction norm for age and size of maturity. And that was kind of a generalized one. This is a bit more specific. It has some kind of theoretical growth curves here. So here is a slow-growing woman, and here is a rapidly growing woman, and this woman is maturing at, say, about 52 kilos, and this woman is maturing at about 57 kilos or something like that. So she's a bit bigger. But this is now data. In the 19th century, according to one measure of maturation, women were maturing at a bit over 20 years of age. And in the 20th century, women of similar genetic background with much better nourishment were maturing more at about 16 or 17 years of age. This is a theoretical reaction norm. These are the observed points. And they were just sliding up this reaction norm up to the left as they got better and better nourished. So I did this analysis. So we asked ourselves, what would happen if modern medicine managed to keep infant mortality rates low? That really started to happen around the turn of the century, and with improvement in hygiene and the advent of childhood vaccination and so forth, infant mortality rates really dropped a lot between the 19th and the middle of the 20th century. The lower curve shows you what's expected to happen. Okay, so the evolutionary response in the entire reaction norm is to drop it down and to the left. Not too much, about 6 months. That's what you pick up. They'll start maturing maybe about 6 months earlier. Okay? The reason they do so is that medicine has reduced the cost of maturing earlier. Remember our analysis over here? We said, gosh, if you're going to try to mature earlier, maybe your babies are going to have high mortality rate. Well, what medicine has done is it's moved this whole curve over to the left, and it's dropped it down a bit. You've cut the cost of maturing earlier, as a result of which the equilibrium point has shifted to the left. And it now actually pays, because of the generation time argument, to mature earlier at a smaller size. That was 1986. Here's data from our analysis of the Framingham population, and this addresses the issue of, is evolution going on in contemporary cultures? And yes, it is. And this is not an actual measurement of an evolved response. This is a measurement of the projected change, given the current selection pressures and heritabilities in Framingham, what will happen to height and age at first birth in this population. Age at first birth goes down by about 6 months. Height decreases slightly, a bit less than an inch. And if you ask yourself, how fast is that rate of evolution? This is for height, this is for age at first birth. These are measured in units of Haldanes, which are standard deviations per generation. And all these other points are all the other measurements that have been made in plants and animals of rates of evolution. And the point is that the humans are sitting right in the middle of that. Okay, the projected changes in Framingham are actually quite normal. We haven't stopped evolving. And not only have we not stopped evolving, but we are evolving, at least in these traits, just the way that life history theory would predict. Modern medicine cuts the infant mortality rate. We adjust downward. We're a little bit smaller and a little bit younger at first birth. Okay, I think what I'm going to do, actually, let me just back out of this, because I want to take some questions, and I don't want to just keep doing PowerPoints. And I want to see how far I am. Okay, I'm almost to the end here. But I'm going to stop because the summary is something you've already absorbed, and I want a few questions. Yeah.
So did the women in the past cut up in earlier and that more time as well?
We,
So are we, so my idea is that,
well,
uh,
chimpanzees did. If you want to call women in the past chimpanzee females, chimpanzees certainly did.
right. So is it, can the trend we observe now be a good thing in the sense that you could imagine that postponing reproduction and reproducing at large size comes with a whole lot of health issues because exposure to whatever comes with postponing things, so that what you just showed us is actually good?
Well, in evolutionary biology, there is no good or bad.
Right.
There's just the facts on the ground. But I have to qualify that by saying that everything I showed you is contingent upon very strong assumptions about culture, right? And I said, this is a projection, and it will only hold if nutrition and mortality rates and so forth remain the same. And my good friend Doug Eubank, who helped with the analysis, said, well, we have to tell people that all we need is one good school lunch program, and that's going to wipe out that effect. So it's nice that it's consistent with life history theory. It's probably biologically real, but it's difficult to project into the future because culture changes more rapidly than biology, and the effects are relatively small. Basically, if what you had going on here was bad school lunches, that effect would be so large that it would cause a bigger delayed maturity if it was a bad school lunch. Not necessarily. That's complicated. I think actually the physiology of that is not reflected in these models. I think the whole business about obesity has multiple causes. You know, Peter Gluckman will talk about some that result from events very early in life, but others just have simply to do with the mismatch between our reward system for eating certain kinds of food and then the availability of a lot of stuff that was never there in our past. So we get addicted to it and eat too much of it. I've been doing it for the last couple of days. Yeah.
I have a similar question. I mean, this is an observation you see of 19th versus 20th century versus 21st. If there was another period of time, like where agriculturists could feed them, and then you notice that we're maturing earlier and it would be lost. How's that evidence of evolution? Just maybe an epidemic phenomenon kicks in when you have good nutrition and you have other things that allow for a change in maturity and changes.
Well, very good question. And if you really want hard evidence for evolution, you're going to have to complete a couple of steps that are actually technically rather difficult to complete. You're going to have to establish the association between particular genes and these phenotypes. In other words, you're going to have a genome-wide association study that gives you a significant amount of variance explained, which is very hard to do. Most of them don't give you more than 1 or 2%. And you are then going to have to follow this longitudinally through enough generations to see the change in the gene frequencies. So you'll have to see that alleles that are coding for earlier age and smaller size at maturity are increasing in frequency in the population. That hasn't been done, and it could be done, and it would require a much longer term and more expensive effort than has been mounted so far. Work on animal and plant model systems suggests that you could probably measure that within 10 generations. So if you started with 100,000 or 250,000 people and you followed their descendants for 300 years, you could probably pin that one right down. That's about the order of magnitude of the difficulty. Yeah. Yeah. It appears to go back to mice, so that's about 50 to 60 million years. That's not a human characteristic. That's a mammalian characteristic, oocytic atresia. So it's had long, let's put it this way, that gives a large number of events in a very long time for the sophistication of the screening mechanisms to evolve. When I forgot to put in that most of the early spontaneous abortions of the concepti, when examined, demonstrate chromosomal abnormalities. So it's pretty clear that one of the reasons for an early spontaneous abortion is a chromosomal abnormality. Well, people have proximate hypotheses, but they don't have proximate mechanisms demonstrated. So one of the things that you would expect, especially if, say, let's suppose it was a mutation in a mitochondrion, and what you were doing was you were monitoring ATP output, and then you were aborting eggs, you were atresia, you were killing off eggs that had significantly lower ATP output or proton flow from the surface of the cell or something like that. I mean, one can imagine molecular mechanisms, but none have been demonstrated yet. It's a tricky one. I mean, imagine getting the material to do the experiment. It's not easy.
Can't really give an evolutionary explanation for fertility transition, the reduction in fertility that happens with modernity. That is, you mentioned the birds and landscapes to reduce the number of offspring when they're adequately nourished. Do you suggest that that might be true?
I don't really think that we can apply the dung beetle explanation to humans because dung beetles have a rather primitive culture. And humans have been using bimodal transmission of information now for a good 250,000 years probably. Bonobos have some culture, but I mean, the problem is that the cultural explanations for reducing family size when infant mortality drops are so powerful that pulling the biological signal out of that one is, I think, non-trivial. You know, the cultural explanation is that often people have many children, either because the men are in charge and they are increasing their reproductive fitness because they're forcing the women to have more children than they want, and there's very good evidence that that is the case. And the other one is that people want to have Social Security in their old age, and they need to have basically bet hedging or risk spreading devices to support them in their old age. Now, both of those are cultural things, and that needs to be filtered out. I mean, one of the themes that will come up in a lot of this is that when we're applying biological explanations to humans, we have to admit that we don't yet have nearly as precise and concrete a handle on gene culture coevolution as we do on the other aspects of just biological evolution. And culture is so important for humans, and it's become such a large part of the selective environment of our biological traits that we really need to solve that problem. And I'm agnostic, because I don't know yet exactly how that works. Carl Bergstrom comes out of a lab where a lot of analysis was done on that. He may have comments later on. Yeah?
Our ability to now manipulate reproductive success, filtering for impact removal?
Well, let's put it this way. It ought to, and building that into the model would be a step towards an understanding of gene culture coevolution because our manipulation is cultural. And of course, there has been a lot of evolutionary concern about assisted reproductive medicine and about whether we are thereby preserving mutations that might normally have been aborted or things like that. To my knowledge, and correct me if I'm wrong, anybody who knows, but the effects picked up so far are so small that they haven't been statistically significant. Peter, I think, has a comment on that.
Show that
Okay, one more question? Then I want about, I have 33 minutes left and I have 45 minutes planned. So, but I can take one more.
Okay, so you said that the presence of the father influenced hormone production?
That's an interesting effect. And
What's the,
what is the effect?
what's the explanation for it? What is it all about?
It is about a move into a riskier environment. And all I want to do now is say that if you compare families in which the father is absent to families in which the father is present, and you look at the ages at maturity of girls in those families, there is a significant difference, indicating that paternal presence or absence is one of the proximal factors affecting maturation. And then there's a whole controversial literature on why. Okay? But the effect is real. Okay, the evolution of aging. Actually, I think this is one of the most important contributions of evolutionary thought to our understanding of the human condition, because it's going to give us some important messages about why we grow old and die, and why essentially the phenotype is disposable and the germline is the object of selection. So this reinforces a couple of points that other people have made this morning. Evolution is not about longevity. It's not about the survival of the phenotype. It is not about the health of the phenotype. Evolution is about making sure that the genes get into the next generation. And if you will get more genes into the next generation, if you have better reproductive performance early in life, then it's worth buying that at the cost of dying. And I'll show you that. Well, aging evolved for the standard life history reasons, okay? Extrinsic risk and intrinsic trade-offs. I'll go through why that happened. I will ask and answer the question, are there any organisms that are potentially immortal? And the answer, I believe, is no. I'll tell you why. Then I'll talk a bit about the mechanisms that mediate aging, and if I run out of time, we'll run out of this section of it. The candidate genes that seem to influence lifespan appear to be general in the sense that you can find them having similar action in mice and nematodes and flies. But when you look at their mutations in humans, they appear to have some particularly nasty side effects in humans. And that actually leads us to look at suggestions of germline genetic therapy for longer lifespan with a bit of skepticism because of the trade-offs that are involved in a longer life. In other words, yes, maybe we could extend lifespan, but we would do so at a price that we might well not want to pay. So, evolutionary theory of aging, and I'm going to give you a couple of quotes from the guys that really got this field founded. So, Peter Medawar, the great Brazilian-British immunologist, said that the force of natural selection weakens with increasing age, even in a theoretically immortal population, provided only that it's exposed to real hazards of mortality. If a genetic disaster happens late enough in individual life, its consequences may be completely unimportant. Let me expand on that for a moment. I want you to think back to when how old your mom was when she reproduced, and how old your grandma was when she gave birth to your mother, and how old your great-grandmother was when she gave birth, and just think about that going back, okay? Then in your mind, I want you to plot a frequency distribution of how old your ancestors were at the point that they gave birth to your genetic lineage. And I think what you'll see is you'll build up in your mind a frequency distribution that has its peak probably in the mid to late 20s. And it tails off as you get towards 40. Okay? Now, what that means is that most of the genes that got into the next generation came from relatively young organisms. And there weren't any that were coming in from really old organisms. And that is the basis of Medawar's statement. And the reason for that is that you start off with a bunch of young organisms in a cohort, and they start to die off. And after a while, there aren't so many organisms in the cohort anymore. And even if they aren't aging, even if they are theoretically immortal, there just won't be as many of them around to have babies. And natural selection is a numbers game, and that means that the force of selection is declining with age. The next piece of thought comes from George Williams, and that is genes that have positive or neutral effects on fitness early in life and negative effects on fitness late in life will accumulate. Okay, this is actually how the cost of reproduction, or the trade-off between reproduction and survival, is produced. It's by the accumulation of genes that are positive early in life and negative late in life. And then George, in this wonderful 1957 paper that you all have in your handouts, said organisms in environments that are risky for adults should age rapidly, and those in environments safe for adults should age slowly. And that's for the reason that I gave you. If you're going to get killed for reasons you can't control anyway, your best strategy to get genes into the next generation is to reproduce and ignore maintaining your body. Okay? So that is where the basic ideas came from. Here's some data. This is actually Brian Charlesworth's graph. And it shows how strong selection is. This is now a normalized selection coefficient. It's been normalized for newborn, and you see that it doesn't really start to drop off until you get into the teenage years and people start reproducing. And what it shows, if you're just looking at selection on mortality rates, selection to reduce mortality rates, that intensity drops to 0 at about age 46 in males in the United States in 1960. So selection pressures are dropping with age. That's what this picture shows. A sufficiently old organism is irrelevant to evolution. I'm out here. I'm irrelevant. Except for my grandson. And that's the point of this. This graph here doesn't account for intergenerational transfers, and that will come up in the discussion of the evolution of menopause. Okay? So, yes, the strength of selection is dropping with age, but when we put in the kind of contribution towards your inclusive kin-selected fitness that can occur when a grandparent is taking care of a grandchild or helping a son or daughter take care of a grandchild, that can shift this curve out to the right and can maintain it at a higher level out towards, say, 70 or something like that. So if you look at the life history theory of lifespan, it says if you want to slow aging, then you reduce adult mortality rates, or you could increase juvenile mortality rates. Both of those will slow aging. That business about increasing juvenile mortality rates may look a little counterintuitive, but basically what's going on here is this. If you've already grown up and matured, and you've survived, and you're a big phenotype, then your probability of surviving another year is good. But the probability that your offspring will survive is getting worse and worse, then there's selection for you to live longer and longer. Because that's the only way that you're going to get genes into the next generation. Okay? So it's not just that you are changing extrinsic adult mortality rates. It's also what's going on in the other stages of your life cycle that will affect how rapidly you age. So if you want to speed aging up, you increase adult mortality rates or you reduce juvenile mortality rates. That's what happened. The reduction of juvenile mortality rates happened in the Mexican dung beetle to the point where, in fact, it only lives 1 year and then dies. And what's going on here is you're altering the relative contributions of adults and juveniles to reproductive success. In other words, conceived of as stages of a lifespan rather than as the whole life cycle. That stage is increasing or decreasing its contribution to reproductive success. And then if you want to have kind of a motto that simplifies this as a take-home, why should you invest in maintaining a body that will be dead anyway? Okay, it's better to concentrate on reproducing while you can. So that is actually about the extrinsic adult mortality rate. If you increase adult risk, then that is how you can think about what happens. So the life history theory of lifespan basically is this. Reproduction early in life is going to contribute more to fitness than reproduction late in life. And it does so simply because organisms die as they age. If in addition to that, there is a trade-off between reproduction and survival, that can increase the intensity of this effect, and it will cause an optimal level of investment in reproduction to evolve. So you ought to put this much in, and that means you will neglect investing or, acting in ways to avoid risk so that you will reproduce. And at that level of investment, the intrinsic mortality will be non-zero. Okay, you won't be immortal. You will have a non-zero intrinsic mortality, and that will determine potential lifespan. This is the idea behind Tom Kirkwood's disposable soma theory. And sorry, I should have checked this morning. This inevitable death is over here, and potential immortality is over here. This is fitness. This is how much you're investing in maintenance or repair. Fitness increases from zero as you start to invest something in maintenance. So you survive a bit, then your fitness is going to go up. At this point, you're starting to experience serious trade-offs with reproduction. It bends over and starts to go down. At some very high level of investment in repair, you might have low fitness because you're not even reproducing anymore. You might be living 1,000,000 years, but you're not having any babies. And Tom's point is that this line, in other words, the amount of investment that you would need to live forever is to the right of the amount that maximizes your fitness. So on this side of the line, you have inevitable death, and on this side of the line, you have potential immortality. That's just a different way of looking at the basic reproduction maintenance trade-off. And I think it's nice because it emphasizes that what evolution tries to do is maximize fitness and not personal survival. So, aging is a byproduct of selection for reproductive performance. It arises by the accumulation of genes that have positive effects on fitness early in life and negative effects on fitness late in life. Is it true? I think one of you sent in a comment about whether or not there was evidence for antagonistic pleiotropy. Somebody do that? I saw that one comment come in. You know, you were asked to send in comments on the lectures. At any rate, there is. You did. Okay, this is the evidence from flies for antagonistic pleiotropy. And maybe we can sample the audience for evidence from humans. I think we don't have anything like this evidence from humans. This is a summary of 14 long-term selection experiments, and they're called correlated selection experiments. Okay, so what you do is you take a bunch of flies and you artificially select the ones that have, say, very early reproduction or a lot of eggs in the first 5 days of life. You do that for, say, 100 generations, and then you ask what happens to their lifespan. So it's a correlated response experiment. I select on one thing and I look at what happened in the other trait. So these boxes are different parts of the fly life cycle. This is their early fecundity, their body size, their late fecundity, their longevity. The arrows are all measuring correlated responses. The sign on the arrow says, if I select for longer-lived flies, they have fewer eggs early in life. If I select for more eggs early in life, this is a negative sign here, they have a shorter life. Okay? And you can see, I think the overall impression that I want you to get from this is that all the parts of the life cycle are genetically interconnected, and you can discover that by doing correlated response experiments. The correlated responses are not always significant. Sometimes there's no correlation. If there's a box with a 0 and a minus, it means that one lab got one response and another lab got another response. So there's something about the lab that makes a difference. And we actually investigated that because we took the flies from the 4 major labs and we went around and we grew them up in the 4 major labs and we measured the correlated responses in the 4 major labs. And it turns out that this is a lab effect and that flies like to live in their own labs and they don't like to live in the other ones. In other words, every fly population is locally adapted to its own lab. So it's evolved to its own lab. Okay? And then underlying that, there's a whole bunch of physiological stuff that's going on that makes a lot of sense. I mean, if they live longer, they're more starvation resistant, they have higher fat content, et cetera, et cetera. You can measure all these kinds of things. I mean, imagine trying to do this with humans. You can't do it, right? But there's substantial evidence in flies and in nematode worms and things like that that there are strong genetic connections between early and late life, and that they are antagonistic in such a way that if you have good early performance, you have bad late performance. Okay? Now, there's been several direct tests of George's second prediction. Low adult death rates should be associated with low rates of senescence, and high adult death rates should be associated with high rates of senescence. So high extrinsic mortality should produce high intrinsic mortality, and low extrinsic mortality should produce low intrinsic mortality. So we tested that. And we did it with a high mortality treatment that should age rapidly and a low mortality treatment that should age slowly. These treatments started off, everybody came from a big population, flies all had the same genetic background, okay, so they all started from the same point. And basically what happened is that we were generating an environment of high risk in which the flies had a probability of living for 1 day after they eclosed, and an environment of low risk where they lived for 6 weeks. So it was a 1 day versus 6 week selection environment. 1 day for high adult mortality, 6 weeks for low adult mortality. After 5 years, they had evolved. So we take them out of that and we put them into standard environments. We measure their lifespan. They'd evolved a 5-day difference in lifespan. And isn't it convenient for your intuition that 1 day in the life of a fly is comparable to 1 year in the life of a human? In other words, if we were to perform this brutal an experiment on humans, and we started at the Trojan War, and we selected them as hard as we could, we might be able to make a 5-year difference in human lifespan over 3,000 years of human history. Of course, that kind of selection intensity could never be maintained in the human population. You can only do it in flies because they have so many babies. Okay? We couldn't maintain that selection intensity. So we got large differences in extrinsic mortality causing a fairly small difference in intrinsic mortality, leading to it's about a 10% difference in lifespan in 5 years. At the same time, weighted eclosion decreased, development time decreased, and fecundity increased. So lifespan doesn't evolve by itself. It evolves as part of a syndrome of the entire life history, and all of these changes are precisely what theory predicts. So you can experimentally, you can carry out experimental evolution. Evolution is proceeding rapidly enough in things like flies and bacteria and worms so that we can actually see the evolutionary process. We can create the conditions that theory says should make a difference and then see if a difference actually evolves. And in this case it did. Okay, now I'd like to quickly switch to the issue of should all organisms age. And this is a wonderful old question. August Weismann asked it in 1882. And he thought that any organism with a soma and a germline should age. He thought that was the critical distinction. He did consider the possibility that organisms wouldn't age because they had symmetrical clonal division. But he didn't really pursue that very much. And then nearly 100 years later, 91 years later, Linda Partridge and Nick Barton had a paper in Nature where they said that any organism with asymmetric reproduction should age. Now, I'll show you what that means in terms of E. coli, which has apparently very symmetrical reproduction. So experiments on a bacterium support that idea, and on Caulobacter and experiments on E. coli suggest that any cellular division is asymmetrical, and therefore all organisms should age. No organisms are potentially immortal. This is the Caulobacter life cycle. It's actually kind of like ours. So they like to live on a substrate, and they have a holdfast where they glue themselves to the substrate, and they have a juvenile stage. So they have a juvenile stage, they grow up, they start to reproduce, they reproduce at certain intervals, and you can actually follow individual bacteria on a slide, and you can count their reproductive intervals, and you can measure their death rate. So you can actually do demography on bacteria. And when you do that, you discover that their reproductive output is declining with age. Okay, this was the first evidence that bacteria age. Since then, there have been more experiments on Caulobacter. There's other evidence, but I just want to put that up and say bacteria age. At least this one does. Now, let's go to E. coli. If you look at E. coli, they have two poles to the cell. They divide in the middle of the cell, and this cell is getting the old pole, and this cell is getting the new pole. And the blue is tracking the new poles, and the red is tracking the old poles. Okay? So the way you do this is by actually labeling the molecules and seeing which ones are synthesized anew and which ones are inherited. If you follow that pattern by following individual E. coli down through a set of generations, what you see is that the ones with the old poles are actually decreasing in their growth rate, and the ones with the new poles are increasing in their growth rate. And the way that they're doing it is through differences in generation time. These guys are reproducing slower and slower, and these guys are reproducing fast. And that is what is leading to this appearance down here at the lower edge of the graph, where some things are doing it faster and some things are doing it slower. This is just summarizing all of those differences in the form of these relationships. So E. coli actually divides asymmetrically, and the old parts die sooner than the new parts. Now, our germline has survived 3.5 billion years. Your testes, your sperm and your ova, and the DNA in your sperm and ova are connected by an unbroken chain of information transfer to the origin of life 3.5 billion years ago. Your bodies will age and die when you're 70 to 100 years old. What an enormous difference. How did the germline do that? Is it because it has really good molecular repair mechanisms? If we studied it really carefully, could we find out how they do it and then use that in the rest of our bodies to try to become immortal ourselves? Or is it because the germline has been repaired at the population level by natural selection by a process that looks like this? We don't know. The two hypotheses are not mutually exclusive. But certainly, this one has been measured and is easily understood. And all it takes is putting the new parts into one cell and sequestering the old parts in the other, and natural selection will keep the young lineage healthy and vibrant, and it will discard the old lineage. And that's going on in oocytic atresia. Okay. What are the mechanisms that mediate aging? The one intervention that works in flies and mice and seems to work in humans is caloric restriction. If you cut your calorie intake 20 to 30%, you'll have a longer life. By the way, that makes you really hungry. 20 to 30% is a lot for most of us. Okay? The mechanism by which that is thought to work is basically that our energy metabolism in generating ATP in our mitochondria creates protons that cause the formation of hydrogen peroxide, both in the lumen of the mitochondria, and they can leak out into the cytoplasm and cause damage there. In other words, there's a very fundamental trade-off between eating and getting damaged. And we are actually loaded with biochemical mechanisms that prevent us from getting this oxidative damage. But if you hear talk about oxidation and the free radical hypothesis, it's those little protons that are the free radicals. My wife always thinks of Che Guevara when she hears the hypothesis, but it's talking about protons. Okay? So if you grow faster and you have a high metabolic rate, that will cause energy flow to increase. And most of the genes that people have been looking for that seem to mediate lifespan are candidate genes in the growth hormone, insulin-like growth factor pathway. And you can either target mutations in mice or you can get naturally occurring mutations in mice disrupting this pathway that will increase lifespan up to 70%. You can get changes in homologous genes in flies and worms that will do similar things. But we're not white mice, and we're not flies, and we're not worms. And I'm sorry for the mission of this laboratory that sometimes model systems don't actually predict what happens in humans, because this particular mutation that increases lifespan by 70% in mice has only been found in one family living on an island in Croatia. They are dwarves. They remain prepubescent, about the size of a 7-year-old, and they're chronically cold and sleepy. And they have no increase in lifespan. Okay. So there may be other target genes that don't have this lack of transfer from model system to humans, and there may be ways that we would be able to repair, say, one genetic problem. But in fact, the evolutionary theory of aging says that aging is a generalized problem. It's not specific to a given pathway, and that in fact, aging will occur by breakdown all over the body. And I think if you've got aging relatives, you know this. You know that different people grow old in different ways. Some of them have problems with their heart, some with their kidneys. Some of them have beautifully tight skin in their 80s, and others are all droopy and foldy in their 60s. And, you know, it's just different for different people. And so even if you tried to go through and using the best molecular genetic technology, fix every single problem that cropped up so that people could live longer and longer, what would happen is that every time you fixed one, another one would pop up. And this could go on for a long time. And when you fixed them, you might be extending lifespan, but it's extremely likely that you would be cutting early reproductive performance. So that's a sobering comment, I think, on the human condition. So I'm just going to skip that and summarize. So aging evolves because evolution favors performance early in life over performance late in life, and the two trade off. That's the evolutionary theory of aging in one sentence. Experiments confirm that lifespan evolves as expected. I showed you a few. There are, in addition, maybe another 20 or 30. Other life history traits evolve with lifespan, as predicted. Caloric restriction is the short-term phenotypic intervention that extends lifespan more reliably than any other intervention. Telomere repair might also extend lifespan. Ashkenazi centenarians have unusually good telomerase and then unusually intact telomeres, the caps on the ends of the chromosome, for people of their age. And germline therapy to extend lifespan seems problematic because there's so many genes involved and because of their undesirable side effects. This has not prevented people from having startup companies trying to market products that mimic the action of some of these genes, but so far none of them have produced effects that are large enough to justify any claims of efficacy or release of the drug. Lenny Warrenti at MIT has started up a couple of those companies, and they haven't worked yet, and I think basically this is the reason they haven't worked. We fall apart in many different ways, and as soon as you fix one, it's a small effect, and then something else kicks in. Okay, questions? You know, if China could actually do that, they can't, by the way, but if China could actually do that, then that would be an evolutionary way to evolve a longer lifespan. And if after everybody in China decided to wait until they were 30, and you did that for 1,000 years, and then said, we're living longer, we can afford to wait until 40, and they did that for 1,000 years, there's probably no upper limit on how long protoplasm can survive. It would keep marching out because the maintenance mechanisms would get better and better as you went through that process. Socially, it's almost impossible to implement that policy. In China, the one-child policy is primarily for middle class in the big cities. There are exceptions for the minorities in the South and for people living on farms. It's actually a policy for about 300 out of the 1.3 billion Chinese. And if you get rich enough, you can buy your way out of it. And so the rich have bought their way out of it, and they have more than one child. Yeah.
So is one implication that maybe you should eat really well during your reproductive years, but then go on a calorically restricted diet because you could potentially subvert holistic sustainability?
Well, that would be one possible reaction. I can tell you my own. I've been the captive evolutionary biologist at a bunch of gerontology conferences listening to what people advise you to do to live a long time. And my conclusion from that is that I would be miserable most of that time. It does really sound unpleasant what you would have to do. And I think that our reaction to this is conditioned by our fear of death, and that if we could accept death as being natural and we can concentrate on being happy while we are alive, we would have lives of higher quality. And seeing that death is inevitable anyway, I think that actually our goal should try to be in as good shape as we can right up to the moment of death. That's every baby boomer's, ideal. And it doesn't work too well because Alzheimer's is going to catch up with some of us and we won't be able to control the process very well. But my conclusion on this is that death is inevitable, and because of our evolved fear of death, which has led to good survival and better reproductive performance in the past, we are placed in a situation that is in fact bad for our psyche. And we'd be better off if we tried to be happy rather than simply avoiding death. I'm not suggesting that we should undertake risky behavior, but I think that there comes a point where there's a trade-off in terms of, it's a happiness longevity trade-off. Yeah. Okay, 1 minute. One more question.
I think you asked the question of whether there was harmful examples of infectious supply proteins. One that I thought of before was the cholesterol problem. There are three simple facts. One is that there's a reason why egg yolks are so rich in cholesterol. We need it for liver development. And statins is a pretty potent sporadically. And patients with deficiency in cholesterol synthesis of cholesterol. Second point is that cholesterol is a very unique compound in our body that there's no way to break it down. You can only eat it. So the importance of cholesterol in development is paramount. And we have very robust pathway to make sure that we have enough cholesterol. Yes, everyone knows about the
Yeah, I think that's a good example, and I think Peter Ellison could give you a bunch of examples with testosterone and estrogen, which both greatly improve reproductive performance early in life and make us susceptible and at risk for diseases of aging later in life. But I think I need to shut up pretty soon.
You can take one more question.
Yeah. Did you have a comment on that?
I just said IGF-1 itself is a good example of really good in early development for child growth, better
Yeah.
So Steve's given us a wonderful overview, and the bottom line for the whole thing is people should enjoy their lunch.
Absolutely. Yeah, don't worry about calorie restriction.
If you'll just follow us, for those of you who don't know the way, across the street, lunch is there. We will reconvene and start right at 1 o'clock.